Welcome to Comet Bio's clinical trial webinar. Today I will be discussing clinical trial results on a gastrointestinal tolerance study that we did on our Arabinoxylan product. My name is Andrew Richard. I am Comet's CTO and founder, and I look forward to discussing these results with everyone today. Before I get started, what I'd like to talk about briefly is prebiotics, just to talk about the basics of them and the reasons why we perform this particular clinical trial. Prebiotics are the fuel to healthy bacteria in everyone's gut. These healthy bacteria lead to health benefits, and fueling them is very important. One of the challenges with prebiotics is that they tend not to be well tolerated, and by that I mean when people take prebiotics, they will have gastrointestinal distress and symptoms. This could include things like bloating or nausea. So it's very important to formulate prebiotics that avoid those symptoms as much as possible. And in our instance, in our Arabina product, which is a natural Arabinoxylan, we wanted to understand what the gastrointestinal tolerance is of that particular material. And before we get into the clinical trial results, let me just talk a little bit about Arabinoxylan and our product in particular. So our Arabina is an Arabinoxylan plant fiber extract. It is a naturally occurring product. It's been very well studied in terms of a general fiber, and it is on the FDA's dietary fiber list. It's found in lots of different plants, leaves, shells of different plants. It's also found in brands, wheat bran, corn bran, oat bran. It is a dietary fiber, and one of its challenges is how it is extracted, which has been proven very difficult to do, which I'll talk about a little bit in more detail. Our particular Arabinoxylan has several benefits, and even before this clinical trial, we had pulled together a large body of clinical research on our product. This has allowed us to have several structure function claims based on the functionality of this material. It is a prebiotic, meaning that it supports the good bacteria in the gut. It also supports immunity. And it supports healthy digestion and healthy normal glucose metabolism and glucose blood sugar levels. So then talking specifically about Arabina in terms of its potency and dose required for a benefit, here we're comparing the prebiotic benefit of Inulin, which is a common and highly utilized dietary fiber, versus Arabinoxylan. And ultimately, we would like to have as low a dose as possible for a given health benefit. So here we're preparing dose required for prebiotic benefits. And that again is helping and proliferating the healthy bacteria in the gut. Our Arabina has an effective minimum dose in and around the 3 gram per day range, which is lower than Inulin, which tends to be higher, although there is certainly some variance based on different clinical trials and literature. Generally speaking, there's a higher dose of Inulin required for prebiotic benefit versus Arabinoxylan versus Arabina. And roughly speaking, we have a benefit at roughly half the dose of other prebiotics. So I mentioned briefly where Arabinoxylan can come from. One of the challenges and one of the main challenges in commercializing this material has been that it's very difficult to extract, it's very expensive, and it's hard to extract it in a form that's useful and commercializable and functionally relevant for structure function claims and for health benefits. In our particular process, which is novel, we actually extract Arabinoxylan from wheat straw, which is left over after wheat harvest. So in this respect, we are adding to a farmer's crop, allowing them to utilize their crop, not taking materials away that would otherwise be used in the food chain, but rather adding to the food chain in a sustainable and upcycled manner. We use a very simple process. We use steam, and we use pressure and we use filtration, and we extract an arabinoxylan of form and function for the structure, function, health claims that I mentioned previously. What we end up with in Arrabina is a material that is highly soluble. It is not a viscous material when in solution, which means that it can be used in a lot of different formulations because of this flexibility and its functionality. So for applications and products such as supplements, nutrition bars, baked goods, beverages, it's a highly functional molecule, can also be used in extruded snacks. And because it's a natural material and it imparts some natural product characteristics, it's highly useful in a number of different applications, which allows us to provide to customers based on what their individual needs are, and then also allows us to work with customers and consumers on lots of different products in lots of different forms. So regarding the clinical trial, what we performed was a placebo-controlled, double-blind, crossed-over study, randomized. 44 healthy test subjects consumed Arrabina at either 6 grams or 12 grams per day, or a placebo. That placebo was maltodextrin. And the setup of the trial allowed for test subjects to take this material, which was formulated into a powdered drink mix. That mix was added to water, 16 ounces, and then test subjects consumed the product over a period of time as per a prescribed protocol. Primary outcome of this study was gastrointestinal tolerance. We measured that by having participants rank on 8 areas of gut and bowel tolerance or discomfort. Things like gas, bloating, nausea, several of them on a 3-point scale. And then we looked at composite scores and then also individual scores for the different instances of intolerance. As a secondary outcome, we looked at bowel habits. We also looked at likability as an opportunity to look at product in this particular format. And we did look at microbiota, and we'll have some data to present on that as well. So again, this was a randomized placebo-controlled study. Each of the subjects saw each of the materials in a double-blinded format. Each test period was 3 weeks in length, followed by 2 weeks of washout, and then the next study. And samples and questionnaires were taken at the start and end of treatment, and then also a daily log was kept by all test subjects. So in terms of results, first looking at GI tolerance results, here is a graph showing composite score. Again, there were 8 criteria ranked on a scale from 0 to 3, which means an overall composite score potential of 24, the higher the number meaning the less tolerant. And here we're comparing the placebo to arabinoxylan at 6 grams per day and arabinoxylan at 12 grams per day. And we can see that there's no statistical differences in the placebo and the treatment arms. And we can also see that the scores are very low, meaning essentially not a lot of reports of discomfort on any of the criteria and any of the scales that we took a look at. So then within tolerance, we wanted to look at bloating specifically. Bloating is a symptom that's very common, commonly reported from consumers who consume prebiotics. Here we're comparing data to Inulin from the literature versus Arrabina from this clinical trial. And we're looking at percentage of subjects reporting bloating as a symptom. And we can see from the chart that roughly over 50% of test subjects consuming Inulin at 5 grams per day reported bloating as a symptom, and significantly less than that for Arrabina at over twice the dose. And this is important because when we think about adding prebiotics to products and adding enough for a health benefit, Most prebiotics require a dose in the 4 to 5 gram range, which happens to be, for some fibers, such as inulin, the point at which bloating and other symptoms can start to happen. So you're requiring a level to get to a health benefit that also causes some distress and discomfort. Contrast that to Arrabina, which is well tolerated at 12 grams per day and probably much higher. But 12 grams per day was what we looked at in this particular clinical trial. And that compares to health benefits at significantly lower doses. So Arrabina can be incorporated into a wide range of materials and a wide range of products at tolerable doses that still provide for structure function health benefits. And briefly, let's take a look at microbiome results. We took a look at the wide Bifidobacterium genus. We did not look at specific strains because we wanted to look at a wide range of microorganisms. Here, we're just looking at Bifidobacterium, and we're comparing the placebo and the 2 treatment arms. And this is a comparison from start of treatment to end of treatment. So how did Bifidobacteria proliferate over the course of a 3-week treatment? In the placebo, we can see that there was, statistically speaking, no differences from start of treatment to end of treatment. However, in both the 6 gram and 12 gram per day Arabinoxylan test treatments, we see statistically significant increases in Bifidobacteria. This supports and is in line with our existing health dossier and adds to the body of knowledge that we have on the benefits of Arrabina. We asked subjects to compare the placebo and the 2 treatment arms in terms of likability. We measured overall likability of the material, ease of use, taste, etc. We saw no significant differences between Arrabina and the placebo, which is a nice result from 2 standpoints. One is this was a blinded study, and so we would like to see that the products were equally liked, and we wouldn't want to see differences, and we wouldn't want to see something interfering with the data. But it also tells us that this is an interesting product and an interesting format to take Arrabina and be able to add it to a beverage. Interestingly, we also asked test subjects open-ended questions around their overall likability of the product. Several subjects highlighted increased water intake as a benefit, and certainly that's a benefit of consuming a material that's added to water. We also measured bowel habits according to a questionnaire. We looked at frequency, consistency, discomfort, evacuation, and straining. We saw no significant differences between Arrabina and the placebo, and importantly, we saw no negatives for Arrabina, even at the higher dose. This is a well-tolerated product, as seen by these data. So in summary, why Arrabina? 3 reasons: gut health, functionality, and where the material is sourced. So in terms of gut health, these data show excellent tolerability of Arrabina. It's something that every customer and consumer is interested in who want to consume prebiotics. We've added to our body of knowledge, which allows for 3 health benefits. Arrabina as a prebiotic, in supporting immunity, and in supporting metabolism, specifically glucose metabolism. It's a functional material based on its solubility and low viscosity, and can be added into a wide range of products. And the source of the material from upcycled wheat straw allows a farmer to use their entire crop. It's a sustainable production of a material where we're adding to the food chain and the food system. And more importantly, it is a novel and innovative product and process in a way to get a useful prebiotic commercially available and looking at a whole host of applications. We're very excited by the product and by these data, and we look forward to continuing on with our research and hopefully presenting more clinical trial results in the future.
All right, now we have a couple questions for the webinar. The first one is, what is the significance of the clinical trial results?
So again, we have an existing dossier of structure function claims. These include benefits of Arrabina as a prebiotic in glucose metabolism and also in supporting immunity. The importance of these results are they add to the body of knowledge, they add to what we know about this exciting product. And gastrointestinal tolerance is a challenge with prebiotics. Every consumer wants to know, every customer that we deal with wants to know what the tolerability is of this product. And we've been able to show that it's a highly tolerable and tolerated prebiotic fiber, which is significant in the existing marketplace.
Okay, we have another question. What's next for the research? Where does this lead you?
So one of the things that we looked at in the microbiota is we looked at a whole host of phyla. And now we'll have the opportunity to take those data and look at specific strains. We know that certain bacterial strains are beneficial, and we know that we would like to be able to proliferate those through prebiotics. And so we will be able to look now specifically at certain bacteria in future studies and how we can tailor this product, if needed, to be able to have benefits surrounding the microbiota. We will also take a look at symbiotics and how these materials interact with probiotics. And then we'll look at other benefits that might be imparted by this product and other health benefits. So we're excited about the future, and I think we've only scratched the surface of what this product can provide in terms of benefits for consumers.
Right, so one last question. What do these results mean for food and drink manufacturers?
Well, it means that food and drink manufacturers can add a prebiotic at a lower dose. Arrabina is very potent, as we've seen from some of the slides, and benefits can be imparted at lower doses than other prebiotics, which gives formulators flexibility. We know that the material is highly tolerated, and we know that it's very functional. But potency is significant because we would like to optimize the amount of material that we add. And consumers are also looking for a benefit around upcycled materials. And this is an exciting product based on how we're extracting it, where it comes from. It's good for growers, it's good for manufacturers, it's good for consumers, and it's good for the environment and the planet.